Hi, previously I showed you that you can use continuous variables, time for instance, for differential expression analysis in limma and HR, and it would be logical to ask, can I do it in DESeq? With this sort of questions you usually go into the user's guide and ask, for instance, for time, and then you see that indeed there is something. You find this small paragraph where it's actually not much said about it, but the reference is given to RNA-seq workflow. So if you go to RNA-seq workflow, in this page you find another user's guide where again you can ask the same question, and then you see number 9, time course experiment. Here authors give example using some data from Fission Easts, where you see pretty standard model using categorical data. There is no continuous stuff over here, and LRT is a completely different story. And you see that they basically tell about using time as a categorical variable, not as a continuous one. But pay attention to this sentence or 2 or 3. Another option would be to model the count as smooth function of time. It is possible to build such a model using spline basis function with R. So it is a trick which we used in limma and HR. Let's try it here. Before I show you my script, please accept my disclaimer. Be aware that here it's quite a bit of a guessing game. First lines are as usual. We import necessary tools and the data. The whole story is briefly explained here, that it's about yeasts which were exposed to oxidative stress, and half of the samples belong to the mutant in a gene ATF21, and then for every time point there were 3 measurements taken in a mutant case and the control case. All the data very well reproducible, and the fact that there are multiple measurements in each time point allows using time as a categorical variable. The trick we learned before is the following. We take our minutes and then import library splines, and then we convert minutes into this magic table X, which will be later used for our model and differential analysis. And of course you can do a poly function as I did in HR and limma, and I tested it here, it also works. Then you have to add these columns to the Fission description, where all other information is stored about data, and you can ask what it is. It is the 3 columns of coefficients. Next I have to decide how I want to use my time, either as a categorical variable, then I take this line, or I want to use it as a continuous variable, then I will have to take this line. In the first run, I will take it as a categorical. I also add strain and replicate. This is all the information known about samples. And then, of course, you can check how big is the data, 36 samples, and what other column data are. As I said, strain, minute scale, replicate, 3 replicates, ID, wild type or mutant, and X, which stands for converted minutes into the spline function. Then, to my surprise, I still have to filter off empty rows. Some gene expressions are total 0. Then I like to use my palette for colors which are numbered, and then I can check a box plot and see, yeah, if it's not normalized, it's no outliers. Then I extract sample information and see that wild type to mutant are 18 to 18, and in minutes there are 6 data points for wild type plus mutants, 3 times each. As it is written, this block about transform time and continuous, so we skip it. For categorical, we use this block, and you must use WALT test, not LRT. LRT has a completely different context. So then we do DESeq, done. Then these are the contrasts. If you're interested in a mutant against wild type, you can run this, but we're interested with the categorical time, so I put together all those comparisons in one using not a contrast parameter, but list. The meaning is that whatever is different between groups, declare it here in these results. And then we go and annotate results with the gene symbols, then remove NAs because some p-values cannot be calculated. Then tops is adjusted p-value below 5% as usual. Then order them and then have a look what's over there on the top. Pretty significant and astronomically low adjusted p-values. HSP9, you can remember this name. Then you can check how many of them, and then you can save tops for later analysis. And if you want to check any of the genes on a list, you take one. This is the second one here, HSP9. And there is a useful function plot counts, which extract count information out of DESeq variable, very convenient. And then you see that indeed this HSP is responding on the stimulus in time, pretty sharp rise and then slightly goes down. The scale is a bit clumsy and line is not available, but you can do it in ggplot style as they suggested in tutorial, so no big trouble. And then it is a beta version of the same, which means it worked, but we use it as a categorical data. Let's go back to here. First we have to declare continuous variable. The input data are the same, but the model is different. You can add replicate. Okay, let's do a strain replicate. We check it again. The sample table is the same, only we use different columns now. We still have to filter. We filter, declared palette, and box plot is exactly the same, why not? But now we run this block where we will use continuous time variable. So these are the columns, and we are interested in time. We put it as a list, and then add annotation column to the result. Check do we have a low p-values? Yes, we do. We skip this block. Now we go to tops. I think I did it already. Yes. And filter exactly the same way, and order, and check ahead. Symbol HSP9. Again, it is one of the tops, which tell us that the guess, and it was my guess, because there was no description how I used spline. Just I based it on my experience with HR. It's okay. We have a 1500, which is a different number, so it's not exactly the same stuff. And then we can save it as spline. And then again I can ask what it is, but you know, you saw it already, it cannot be different. it's the same stuff. What we do now, because we have two lists, I call lists previously saved for continuous and categorical time variable. Then I call Euler ask for when and plotted. And then I see that intersection is pretty rich. Continuous variables gives you less time-dependent genes. What is curious in this case that log-fold change stays in the results in n-tops table in DESeq2, unlike to limma and HR, because they still report log FC change of time value compared to the 0. So it's still present, but you have to take it with correction, what it means. And then to make sure that it is significant overlap, you have to know the total number of genes, I take it, what we have in the initial data, the size of the overlap, what is continuous group, what is categorical, and then use this nice reference to Stack Overflow, where Ferdinand Kraft, unknown to me, wrote a very elegant function to calculate enrichment value for the Venn diagram, and it says, yeah, if I didn't use replicates, it was this value, and with replicate, it is now this value, which is pretty significant anyway. And for those who like to calculate these enrichments, you can go and check RVen package, which claims to calculate significance, but it didn't work for me. There is also Giant package, where significance of gene lists is calculated, and there is also p-hyper function, which is referred in this Stack Overflow reference in this script, which is traditionally used for enrichment, but this function is different. It's not based on a p-hyper. To conclude, it works in DESeq, although you have to figure out many lines yourself. The user's guide is not very helpful in this case. If you are really interested into the genes induced by this oxidative stress, then formally speaking, you have to do some sort of clustering and classifications of these time profiles. For this, I simply copy-pasted lines from the previous limma and HR protocols, which I mentioned at the beginning of this video. It's not an ideal solution, but it works to some degree, and I can repeat it here briefly. So you do a call of the library, for some convenience also define the colors, then you extract counts out of the DESeq variable. Then you can check how big are the changes between the maximal and minimal expression values for any gene in your list of 1,000 significantly changing genes. Most of them do not change much, and therefore I prefer to cut it somewhere above 3. It's log values. I will analyze only a bit more than 200 genes. That's more than sufficient actually. Then I generate MDS plot by correlation. Then I employ H-class to cluster all these groups of genes and divide it traditionally on 9. It's a bit excessive, but hopefully sufficient. And finally, I can visualize all the 9 profiles of all the clusters by defining scales, finding my minutes back. Then I still have to split, either I want to see wild types or mutants. It's a bit messy if I show it at the same time, so S is my filter here. Let's do it for wild types. And then we have those 9 different profiles. Of course, they are a bit redundant. Cluster 1 contains HSP 9, going up then down. Some genes go down then up. That's more or less what you're supposed to do, and do it better at the end. And after that you can also go for gene ontologies and other characterization of the genes. This will be really the end of this differential analysis.